Good evening to all the participants. Welcome to the day 3 session on methods of drug design. Before proceeding to today's session on methods of drug design, can we recap the last session topics? So we can discuss the session topics at present. So what exactly did we discuss in the last session? Yeah. So just either unmute and answer, or you can type the same in the chat box. Okay. How about the others? Target identification, convolution, deconvolution, protein structures. Target identification, if you see how to download the 3D structures of proteins from various databases that we have discussed, isn't it? So can you mention me the structured databases for targets? So what are the structured databases for targets? So we can discuss one by one. Yes, we have discussed even on protein structures, level of protein structure predictions, and then we have discussed on methods of protein structure prediction. You're right. So I'm just asking you the structure databases for targets. No, iTaser is the tool for developing the structure through computational method. So this we are using threading concept. We are using threading to develop a protein structure. Still, this is a tool. Databases, you remember? Can I give you a clue? PDB. Like PDB, what are the other databases we have discussed? Where we can download 3D structure of proteins. Other than PDB, so PDB is from RCSP, isn't it? As a Center for Structural Collaboratory Protein Data Bank. Discovery Studio is a visualizer to visualize the protein structures. But if you want to download the data of 3D structure, you have to download from database. So Target DB, no, still not. CHEMPL for chemical compounds. If you want to download 3D structure of chemical compounds, you can use that. And if you want to know the target information of the chemical compounds, you can use Target DB. Modeler is a tool. UniProt is a database. UniProt is a fully annotation database. So here, majorly, we can get information of sequences as well as we have structural information through hyperlinks. It directly direct you to the hyperlinks. So if you see this, this is a fully annotation. You find sequence and structure. Can you give me more specifically, other than PDB, what is the structure database present in NCBI? In NCBI, we have a 3D structure of protein database. What is that? What is the name of NCBI structure database? Can someone tell me? Abbreviated word? Not exactly. You are very close. GenPept is for protein sequence, but for structure, yeah, it is MMDB. And very good. I appreciate all the participants interacting effectively from day 3 session onwards. And after the end of this third session, I'll be circulating you the feedback. Also, provide me the feedback for the past 3 days. How was your experience? Okay. Oh, very good. So you're right. Very close, but one is PDB, the other is MMPDB, and if PDB is from US database, MMDB is also a US database. From Europe, we have a database called EPDB. So this is also used for downloading 3 structures of protein. So these are the databases. And now the next question is, um, give me some examples for homology modeling servers. So what are the examples we have discussed? Very good. Swiss model. Anything else? You remember? Okay, but even the Swiss model is sufficient enough for us to practice. At least we should remember one kind of model. Anyhow, I'll be showing you just for a recap. So if you see the homology modeling servers, 3D jigsaw, CPH models, ESY Pred 3D, Geno 3D STC. So many of them try to use the Swiss model. Okay, the Swiss model is the popular tool, of course. Like this, we have threading. We have discussed on PHYRE, and ab initio, we have discussed these tools. Okay, so this you can get from Expassive Web Server. Can I go ahead with the next question? So the next question is what are the five levels of protein structure prediction? Protein structure. What are the five levels of protein structure? Very good try. We have discussed only on Swiss model and PHYRE. We have shown that. Primary, secondary, super secondary, tertiary, and quaternary. Excellent, excellent. Okay, so all this information we got in a single answer. Good. Next, can I go ahead with the next question? We have discussed on Ramachandran plot, isn't it? So in Ramachandran plot, we have we are concerned with the two amino acids. Majorly, we concerned with two amino acids in Ramachandran plot. Can you tell me what are the two most important amino acids which we concern in Ramachandran plot? I told you Ramachandran is Indian bioinformatician. He is also called as father of bioinformatics. Very good. The answer is glycine and proline. You know why we come across glycine and proline? I also mentioned you related to alpha helices. So the glycine and proline are poor formers of alpha helices. If these are poor former of alpha helices, these are used as an indices to distinguish whether the amino acid sequence is forming alpha helix or beta structures. You know, in at existence, you can see more than 52 to 53 percentage of proteins have alpha helix structures. So the remaining 47 percentage of the protein structures have beta sheets, loops, turns, coils like this. Okay. So alpha helix contributes major of the protein structures, more than 53 percentage of the total protein structures available. So one should concern about this glycine and proline residues. That's what you can even see in the Ramachandran plot. If you see this Ramachandran plot, the Swiss model, okay, yesterday whatever we have generated, okay, these results will be in the repository for a week. And if you see this Ramachandran plot, if you want to download the Ramachandran plot, I told you to click structure assessment. So this is an output of Swiss model. So just because I fed in my email ID, I have received this result. This result we can see up to a week. And here, if you see in this Ramachandran plot, like however we find phi versus psi plot, okay, we also this is found in general Ramachandran plot. So whatever 706 amino acids of myostrap you can able to see it here. For example, if I click here, you can see this is 309 39th position. This is unfavorable residue. And if I keep somewhere here, okay, you can get the position like arginine 29. So like this, each and every amino acid is represented by a dot in this Ramachandran plot. And as I mentioned you earlier, the glycine and proline residues distinguish the alpha helix, the major from a major secondary structure alpha from beta sheets. So we also have a separate Ramachandran plot for the glycine residues alone and proline residues alone. If you have already worked, you may have this kind of an knowledge, but somehow I am showing you just because I'm asking you this kind of a question. And as I mentioned you on earlier, the Ramachandran favor regions, if it is 80% and above, we can keep the protein quality as good and we can take it further for docking simulation purposes. If the Ramachandran favor residues or less than 80 percentage, I suggest you to not to use the structure. But in some journal papers, you can see above 50% they take even for any other drug design applications. But that is not suggested. Okay, you can't able to answer for each and every reviewer. Okay, so they won't accept all these reasons. So better go with 80 or 90% above. Okay, and we can tell the stereochemical quality of a protein is good. Okay, this is what we have discussed, and we have also discussed one server to assess the protein quality. What is that server called? Can I give the abbreviated name? Project, very good. Project is also used, and CASP is also used. So, what is the expansion of CASP? Actually, even some computational prediction servers like project can give bias, but this kind of a bias is corrected in a kind of an algorithm proposed by Murray, I believe. That is a scientist. So critical, very good try. Critical assessment structure prediction. Critical assessment structure prediction. Okay, so that's the expansion. Good try. Now, let me go ahead. So, I think target validation, target deconvolution, we have seen. Only thing is that we have to see the practical examples, how to use the tools, how to use the databases for whatever we have discussed. Okay, now let me go ahead with the session 3. Okay, so let us start with session 3. So, a lot of things to recap. So, today we'll be discussing on types of drug design, drug discovery and development, and we discuss various computational methods of drug design. So, if you see in earlier phases, this kind of a statistics taken 15 years before. So, 15 years before, for completing the computational studies like target discovery, screening, lead development, they have took around 6 years. Okay, 15 years before, we have to take 6 years to complete the computational studies. But the when we face the COVID-19 pandemic situation, when it is declared on April 2019. So when it is declared in April 2019, on June 2019, on June on June 2019, so it was we got a kind of a journal paper from University of California that a scientist has docked around 1.3 billion molecules. 1.3 billion molecules. Okay, that is whatever repurposing compounds present in the database. He has docked all the 1.3 billion compounds with the spike glycoprotein, which is the major target of SARS-CoV-2. Okay, it's see whatever earlier 15 years before they work for 6 years, he can able to make it within a short period of time, within 1.5 month. He can able to even publish the kind of a data of whatever he has done. So what he used is a supercomputer in his university, and he made this possible within few months itself. Okay, and if you see, like even whatever computational studies used for bringing a vaccine other than the drugs, okay, even it took not more than 2 to 3 months. Whatever Covishield, Covaxin, and other 87 types of vaccines discovered around the world in 2000, in September 2020, okay, around 89 vaccines in the world came for the prevention of SARS-CoV-2. So, but we know only the two types of vaccines, isn't it? But the computational studies for this are already completed within 2 to 3 months. Only thing is that to check the efficacy of the drugs or vaccines, we have to first try with the preclinical development. That is checking with animal trials. Okay, so that's what we call it as a preclinical development. Definitely, I think you know that the PhD scholars, if you work on animals, minimum it took around 4 to 6 months, isn't it? Minimum 4 to 6 months. It's not like zebra fish or some other kind of a worms you can able to complete within 1 or 2 weeks. So for preclinical development, we are trying with small animals like rodents, mice, etc. So obviously from 2 to 3 months, and then 6 months for doing with preclinical, so 9 months, and then next what we have to do, we have to go with the phase one, phase two. Phase one we try with 100 healthy volunteers, and in phase two we try with 1,000 healthy volunteers to check the efficacy. Obviously, it took around even 6 months, 6 months separately. So within 1 year, so yeah, minimum we had 1 year, 9 months to bring a kind of a vaccine. But less than this period, we got 89 vaccines, and so many drugs came into the market for the prevention and treatment of SARS-CoV-2. Okay, so the rapid development has happened in the drug discovery and areas of modeling and simulation. In bottom, you can see what are the different computational methods used in modeling and simulation. Now, let me come across the types of drug design. The first type, what we discuss here, is the traditional drug design. That is, before 50 years, how the drugs are discovered. The drugs are generally taken from the natural sources like plants, animals, algae. So, whatever they take, they grind it using the pestle and mortar, and the mixture of compounds. Even sometimes they use a mixture of 3 or 4 plants, or sometimes 20 herbal plants also available, isn't it? 20 herbal plants in the form of a healthy mix, or a kind of a remedy, a natural remedy. So like this, we have so many kind of things that developed from Ayush department. This Ayush, a separate department. Even now it is recognized by BIS, and they also have their own kind of department to support this Ayush. So if you see a combination of herbs, so everything, and so you have in one plant itself, you have more than 200 compounds. And if you take 20 herbs, think about the number of compounds. It's We have more than 2,000 compounds, but not all the 2,000 compounds can able to treat a particular disease, whatever we are trying to. So then what exactly we have to make? So we have to initially try with structure based drug design. So recently, that is past 40 years, we are working on the structure based drug design. So if you see in this 4 decades, so the past 3 decades they are trying well, and if you see in this past 3 decades, the natural ligands are screened initially, and here we need the 3D structure of both the ligand as well as the target. That is, if and if you know the 3D structure of a drug and 3D structure of a protein receptor, then we can able to at reduce the price of manufacture of the drug. If you want to produce the drugs, obviously first we can try with the computational studies, so that even the investment in vitro and in vivo studies will be less. Okay, so first so for completion we need the 3D structures. So first we should model the structures. If the structure is not available, then we have to go with docking, simulation, etc. So more about these methods we'll be discussing in the subsequent slides. Okay, so this is what structure-based drug design. Obviously, after completing the computational studies, and we go with the synthesis of compounds. So for synthesis, we can go approach the medicinal chemistry division. And if you're going for high throughput screening, we can also use combinatorial chemistry division. And whatever compounds we synthesize, we can first check with in vitro, that is checking through cell lines. Later, we can check with the in vivo in small animals. Okay, later, after completing the in vitro and in vivo successfully, we can go with clinical trials. Drug discovery and development. So if you see the first step, what we go ahead is the target discovery, which is a strategic research. Here, these are the three kind of protocols we follow. That is program selection, target identification, and target validation. That's what we have discussed on yesterday. And the next is exploratory research. Here, we are focusing on ligands. Here, if you're focusing on target or receptors, here we generally focus on the ligands. I told you earlier, the ligands are usually drugs in small molecule discovery for the treatment of diseases. But even if you see the cancer like diseases, we majorly focus on antibodies, isn't it? So antibodies, and even a DNA binding protein, RNA binding protein. So we use a different thing, peptides, lipoproteins. All these things can also be used in the form of ligands. But if you see a small molecule as an example, initially we go with heat generation, then we go with the lead identification. Okay, out of numerous similar compounds, we try to find which is the best compound. That is what we call it as a lead. So whatever we identified as the best compound lead, we try to optimize it. Like we detach the functional groups in the existing drug, and we attach with some other functional groups, and we check whether the binding efficacy is going to increase or not. So to optimize the lead, the existing drug. So actually in pharmaceutical industries, we have quality control and quality assurance. Quality control and quality assurance. So what is the purpose? Why they should have an R&D division in the pharma? Like whatever drugs they synthesize, generally if you want to check the quality, especially in terms of purity, yield, they try this. But even if they want to develop anything new, they can also go with a lead optimization protocol. Okay, and whatever they develop the leads, that's what we call it as a new investigational drug, IND, investigational new drug. If they bring a kind of a new drug, we call it as an investigational new drug or new drug application. So this will be checked with animal models, and then try with the clinical. And in clinical trials, we have three trials. Phase one is for 100 healthy volunteers. Okay. And the phase two, we try with the 1,000 healthy volunteers. And in phase three, we try with more than 1,000 unhealthy volunteers. More than 1,000. Okay, so like generally we want to try with the patients, isn't it? So we try with all these three clinical trials. Okay, stepwise, like if and if it clears a phase one, we go to the phase two, and if the drug clears a phase two, it can go to the phase three. Many of the drugs, even clearing the in vitro studies and in vivo studies, fail to clear even phase one. Some drugs can also clear up to phase two. Okay, that is up to healthy, there won't be any problem. But if it's treated to the, it's taken to the next stage, phase three, it may give some kind of a side effects, or it won't give a better efficacy. So under that case, you can tell it is a phase two drug, not a phase three. But if it even clears a phase three, obviously that phase three cleared drug comes to the market, and even that kind of a drug can be applied for Food and Drug Administration Act. Okay, so the FDA approval. So FDA is from US. If the drug clears up to the phase three trials, the drug can we can that drug manufacturer can apply for US FDA, or he can also apply for EMA, European Medical Agency. So we can try any of this based on whether they want to export to US or European countries. Okay, and this is how you can see these are the stages or phases of drug discovery and development. And if you see in drug discovery, we have so many nearest terms: drug design, drug discovery, drug development, drug illustration. Okay, drug design is not mentioned. You can take it as a drug design. The drug design is what we'll be discussing in this chapter. If you see this drug discovery, okay, so here generally we can try with the clinical trial, I mean the in vitro studies. Okay, we get a new targets, we can clone the targets, and then we go with the biological assays, and then we also can link with the drug design. In the drug discovery itself, here they are also linked with the drug design. Okay, and if you see in the drug development, whatever drugs we have, numerous drugs we have, we screen a base drug and we go with the lead optimization. That is, start from the hit to lead generation, and lead generation to lead optimization, and then we go with the pharmaceutical scale. Like if you see SARS-CoV-2 patients are gradually increased from 2019 to 20, isn't it? So many patients are also died just because of the multiple illness factors. If so, then there is a need to even get more amount of drug. So pharmaceutical scale up related to that, and checking the pharmacokinetic properties in small animals. Pharmacokinetic properties or absorption, distribution, metabolism, excretion, and even safety and toxicity studies what are checked with the small animals. And it later it can be formulated to tablets, capsules, syrups, ointments based on what applications we make. And then obviously whatever applications we bring, whatever formulations we bring, we have to try with the clinical trials, phase 1 to 3, and then phase 4 is linked with the FDA approval and post-marketing surveillance. And drug illustration, actually instead of trying with small animals, we can also try with nematodes, worms. The worms can serve as a model organism for human. I don't know how many know this. Okay, and even the zebrafish, the zebrafish size. Okay, the fish is having more than 60 percentage of analogy with the human genes. So worms, fish. I think zebrafish may even know. Did you heard about this Drosophila fruit fly in the banana fruit, in the ripened banana fruit? You can see so many fruit flies, isn't it? Even that Drosophila can serve as a model organism for human. Like if you want to check for treatment of any diseases, so these worms and Drosophila can be used as organisms, model organisms, and even the synergistic effect and the other kind of an effect. Okay, the adverse effects, everything we can try with these kind of an organisms also. Okay, and related to the genes, genetic disorders, everything also we can try with this. Okay, so something special, isn't it? Generally, whatever we carry is drug discovery and development. Drug illustration is something peculiar. We have to check with the type of disease whether we can try or not. Now let me go ahead with the computational methods in drug design. So there are so many methods in drug design. We can see the most important methods. First is drug likeness prediction. We have tools, open source, I mean free online tools. Go to the server, just upload 1D format of a chemical compound. The 1D format of a chemical compound is generally a canonical smiles format, is a canonical smiles format. So we have to feed this as an input and we will be getting the drug likeness. More about the drug likeness and ADMET prediction we'll be discussing on tomorrow. Okay, so we have a separate session for this. These are the tools, online tools, freeware tools. And for QACR, quantitative structure activity relationship, quantitative structure property relationship, we use this kind of a tools. And whatever I highlighted in blue color is a commercial tool. The remaining are all freewares. Okay, and pharmacophore modeling, we can use PharmaGist and PharmaMapper. Molecular modeling, we have already discussed on yesterday. Molecular docking, okay, we have so many tools for docking. Thousands of tools are available. We can see Autodoc as an illustration tool for molecular docking. And inverse docking, we can go with ACID. This is also an online tool. So we have molecular docking, reverse docking. The reverse docking is what we call it as inverse docking. Okay, we discuss about that also later. Molecular dynamics, whatever we call as MD simulation is what molecular dynamics. Okay, GROMAX and NAMD, and read across approach, that is checking toxicity, toxit, all tox read, tox spread, like this. These are some of the tools. And for system biology approach, we can go with gene expression profiling and biomarkers development. So generally for seeing the protein-protein interactions, DNA-protein interactions, ligand-protein interactions. So all these kind of interactions we can see using the freeware visualizer called Cytoscape. Okay, of course we have, you're going to use plenty of databases, and from that gene expression databases, you can download the data in the form of data sets. And from the data sets, we can able to find the profiling. Similarly, for the biomarkers, we have plenty of databases. So, of course, we use the tools to analyze the kind of these results, gene expression and biomarkers. Virtual screening via high throughput screening. Like if you want to screen multiple drugs and you want to screen the best drug for treatment of your disease, we can go with multiple virtual screening tools. One such popular virtual screening tool is Autodoc PYRX, which I'm going to provide you a demo in one session. And the machine learning techniques, we have machine learning techniques like DeepMinder, Python programming for working on inactive compounds. QSAR, quantitative structure activity relationship models. Okay, actually, here A stands for activity, and QSPR is the general term, that is quantitative structure property relation. So, in this property, if you are studying on activity of compounds, we call that as QSAR. Okay, QSAR is a part of QSPR. And similarly, if we are working on reactivity, okay, reactivity relationships, some compounds are having the reactions, showing the reactions, isn't it? Especially even in enzymology, if you so you know that ligand and receptor, if it binds, it is a reversible reaction. Ligand receptor complex is formed. Okay, so and if you keep this as k1 as forward reaction constant, and this is a backward reaction constant. But if you come across in the case of enzyme and substrate, okay, if you use a substrate as a ligand, okay, like a TMB, and enzyme, any particular enzyme, of course, this is also a kind of a reversible reaction. But what it happens later, it going to this enzyme substrate complex, whatever form, is going to dissociate into enzyme and product, and the rate constant can be given as K2. So generally, the ligand receptor, we find only ligand receptor. So it is a kind of a reversible reaction. But in enzyme substrate, it's something you find a peculiar thing. Like after forming this intermediate complex, which is unstable, it can degrade it to, it can further react to form enzyme and products. Okay. So this is how the dissociation happens. And this, of course, K2 is a dissociation rate constant. Okay, so the reaction is happening with this substrate, the ligand, isn't it? So there you should have knowledge on QSRRs and QS chromatography relationships. You know, myself, I'm working on a series of plant compounds called methanoloids. Methanoloids or steroidal lactone compounds of ashwagandha. Okay, as the name itself implies, steroids, plant-based steroids. The plant-based steroids show less kind of a side effects when compared to animal steroids. So I'm trying with these kind of effect of these compounds for the various diseases. And if you see in ashwagandha, we have around 41 methanoids. Each and every year they are increasing the number of ethanoid structures. Okay, so when I started research, nearly 35 ethanoids are there. Now it is 41, 42, 43. They're increasing, so approximately 41 ethanoids are evaluated or found in ashwagandha. So if you see that all these 41 ethanoids have more similar kind of a molecular formula, molecular structure is also more or less same, size, etc. So it's very difficult to even to separate it using the chromatogram. So under these kind of a complications, okay, a series of closely related compounds, we can also use quantitative structure chromatography relationships. And for assessing the toxicity, we can go with the QSTS. Okay, and for assessing electrochemistry, like wherever you find the corrosion related thing. Okay, so whether the based on oxidation, redox potentials, one can go with the evaluating of the quantitative structure electrochemistry relationships and quantitative structure biodegradability. This is what we generally study in environmental science and engineering, isn't it? So, and if you see these kind of ways here, so basically we should understand that whatever structures we have, okay, we should have initially have to check the experimental techniques through in vitro experiments. Later, we can also bring in the form of a molecular descriptors, and we can check the kind of a correlation between these two using these tools. Using a machine learning technique, we can able to make it. And related to this, we'll be discussing some more in the subsequent sessions. Okay, and the softwares used in QSR is mentioned here. Paddle, OChem, QSRINS, Minitab, Alva Descriptor. Of course, Galaxy is not there. Galaxy is also one of the tool. Reverse talking, the picture quality is not that much good. I can explain you somehow. In docking, we have only one target. Okay, but we have multiple ligands. If we have a multiple ligands, and if you want to screen the best ligand, that is from heat to lead, if you want to bring a lead, so we can try to talk all these compounds and check which compound from 1 to 10 compounds if you have, which compound is going to show higher negative energy, which is going to show negative maximum energy, that we can tell later. Uh, we can we can assess that as the best compound, best drug for the particular target. You know, in Autodoc, that is a manual docking tool. In this Autodoc manual docking tool, we'll be getting scores in terms of kilocalorie per mole. Okay, if we get negative maximum energy, for example, if second compound is showing more negative maximum energy when compared to the remaining nine compounds, we can judge that, we can finalize, we can confirm that the second compound is the best drug for this. So this is what usually we carry the talking process. But in reverse talking process, instead of trying with a single target, we try with the multiple targets. Okay, we try with the multiple targets. So one compound we try. So what is this actually? Did you hear this term already? Or whatever we discussed in earlier thing, like target deconvolution. That's what we here we call it as a reverse talking. Okay. So, but obviously one ligand, we try to talk with multiple receptors, but obviously we need a best kind of a protein. Okay, so that for a particular disease, so we rely on one particular target of a particular disease. So we check which compound is binding perfectly to that particular target in the reverse talking. Okay, this is a kind of a target deconvolution approach. For this you can use acid. So under this acid tool, so we have to just upload a plant compound, any kind of a drug, okay, or a plant compound in SDF format. SDF stands for structured data format. So the structured data format is a 3D structure of a compound. If you can able to download a 3D structure of a compound from NCBI PubChem, this is the database. Okay, and after downloading it, if you upload this as such, and if you run in this URL mentioned below, okay, so you can able to see the series of protein receptor targets. Okay, so the vitafrin A, whatever I have uploaded, this is a best oxidoreductase inhibitor. So according to target deconvolution, how we can conclude the result. So what this is a kind of an output result we have received. So oxidoreductase, you can see it is targeting more than 20%. Next to oxidoreductase, we can also find this plant compound also as a transferase inhibitor, and also it is a hydrolase inhibitors. Okay, so it's also showing the activity for the other kind of receptor mechanism like ligase, transport signaling ligase, but this shows something less when compared to the top three receptor activities. So this is what we call it as reverse talking or target deconvolution. So next we come across with molecular modeling. I think we have discussed extensively on yesterday. So after doing homology modeling, threading, ab initio, we can compare with comparative modeling. The comparative modeling is comparing the computational predictor structure from any of these three methods with experimental structures. Okay, so obviously this can give you a kind of a better kind of an assessment of whatever structure we have predicted. Of course, we also use the Ramachandran plot, and from Ramachandran plot, number of favored residues can give you an idea on the stereochemical quality of a protein. Other than this, we can also assess the quality through CASP tool, and RMST, root mean square deviation, can also be checked for the protein to assess the quality of the proteins. Now, let me take you to the molecular talking. So this is the next protocol in drug design, most important protocol in drug design, because in structure-based approach, we start with the docking-based approach. Of course, we have also pharmacophore modeling, a hybrid-based approach, but the more significant is the docking. So if you see docking, as I earlier mentioned, we have two important terms. One is ligand, the other is receptor. One is ligand, the other is receptor. So whatever we have these two terms, the ligand is a small molecule and receptor is a large molecule. Okay, we try to check the binding affinity and we measure this through energy. How much efficiently a ligand is bound with the receptor, we assess through the binding energy. I told you in autotalk we measure through kilocalorie per mole. Okay, so one thing you can see that docking is a method which predicts the preferred orientation of one molecule to a second. This is first and this is second. When bound to each other to form a stable complex. I told you, no, the ligand and receptor intact to form ligand receptor complex. Knowledge of the preferred orientation in turn may be used to predict the strength of association of binding affinity in terms of energy. So, how to, if you see in Autodoc, we have only binding energy. But if you see a commercial tool called Ligand Fit, the Ligand Fit is a tool from Discovery Studio. In Discovery Studio is a package where we have numerous programs for drug design. Ligand Fit is generally used to dock a single ligand and a single ligand with a receptor. Okay. And if you try to dock a single ligand with a receptor, we can get around eight different scores. Eight scores, not just an, we don't have any energy term, but we have scores like PLP1, PLP2, chain, okay, and MMF. We have so many kind of scores. So whatever scores we generate is from DS ligand score model. DS ligand score. So DS ligand fit is the program to dock a single ligand and receptor. DS ligand score is another module which can evaluate the binding affinity and it shows the result in the form of eight different scores. Each and every score is a combination of physicochemical parameter of the ligand as well as the ligand receptor complexes. For example, pK of a ligand and molecular weight of a ligand, we used to calculate score 1. And the pK of a ligand and hydrophilicity of the ligand can be used to calculate score 2. Like this, a combination of physical-chemical properties are used to calculate 8 different scores. Then you can ask me the difference between the Autodoc and DS ligand fit. Autodoc, we have only one energy. So the precision of checking the binding affinity is less when compared to Discovery Studio. So only the Discovery Studio, even if you want to procure, it will be in lakhs. Okay, so it is in lakhs. Obviously, just because we have more advanced algorithms developed and also more not only for the scores calculation, but also for binding sites prediction. And even we have a few more efficient algorithms to check binding cavity prediction, etc. Okay, so like this we can see, but the most important thing in molecular docking is search algorithm and scoring function. This is how a commercial software and freeware distinguish with each other. And if you more about this, we'll be discussing in one session, I think on day 5 session. And the assessment of docking can be measured like this. And this is a kind of a peptide docked with a protein. So this structure you can see, this is a peptide docking with a protein. And we have thousands of tools, but this is the tool I'm going to provide you the demo, Autodoc, and I'm going to explain you with the Discovery Studio Visualizer for analyzing the results. And the next is the molecular dynamics. Molecular dynamics is a computational simulation method for analyzing the physical movements of atoms and molecules. Like if you want to check the type of interactions, okay, we have two types of interactions: bonded interactions and non-bonded interactions. So using these bonded interactions, you can able to see there are three bonded interactions available. Whatever you can see, this atom is stretched from this part to this part. I mean, this is the stretched part, isn't it? Some small part is stretched. So whatever we calculate is the bond stretch energy. And here you can able to calculate the bond bend energy. That is before bending and after bending, you can calculate it. And this is rotational along bond. So that is rotating along the torsion axis. So we can able to calculate the moment of inertia from this. So the sum of the these energies, we get a total bonded energy. And non-bonded energy can be calculated as the sum of the van der Waals energy and electrostatic energy. So the summation of bonded and non-bonded energy is what the total energy of a protein. If you see a protein, you have numerous atoms. First, you have a numerous molecules. For example, if I go with the trap one mouse, we had 706 amino acids. And if you see the atoms might be 1700 or 2100, something like that. The atoms definitely more than this. Okay, definitely more than this, even up to 10,000, something like that. So if you have this much atoms, okay, so how these molecule and atoms, okay, have this kind of an energy. So for each and every molecule, this amino acid, that's what I call it as a molecule. For each and every molecule, we calculate energy through trajectories, energy trajectories, and we sum up all the energy trajectories of 706 amino acid to calculate total energy. And then whatever energy we calculate, we go with energy minimization protocol. So there is another method of drug design called energy minimization. So for energy minimization, so for initially, if you go with a protein, for a protein, we have to calculate for each and every amino acid molecule, and we sum up all the energies of this amino acid molecule, it which is nothing but a summation of bonded and non-bonded energies. So that whatever energy we calculate, we go with the energy minimization protocol to make a protein more stable. To make protein more stable, we go with energy minimization protocol. Okay. So if you want to check the effect of solvents on a particular protein, instead of trying in lab experiments, we can try the same even in computational experiments. And also, whether a ligand is bound with a receptor, okay, or if you want to check the binding of ligand with a receptor over a period of time, we can go with molecular dynamics simulation. So again, I'm telling you, if you see in docking, we generally assess the binding affinity of ligand and receptor. The same binding affinity of ligand and receptor, if you want to check over a period of time, that is from 100 nanoseconds, what is going to happen with a drug? Okay, if a drug is going to bind, and one thing you should remember about this, I can come up with general examples instead of providing you more generic terms in computational method. So I can ask you for we can discuss few things in the form of questions. Yeah. So my first question is, what is the drug we use to treat common fever? So then I can link with the computational methods. What is the drug we use to treat common fever? This is my question. Paracetamol. Paracetamol is not a generic name. What is the generic name for this? Acetaminophen is the generic name. You are right. And you know how many doses we have to take per day for a common fever? How many doses? And what is the dosage? Two doses? No, no, definitely. Yes, the three is right. Okay, and depending upon body weight, we can go with like a 50 kg and below. The doctor prescribe around 500 mg, and above 50 kg, the doctor prescribes 650 mg for three doses. If it is two doses, it won't cure. Okay, three doses is standardized, and 500 to 650 based on the adult's weight. But if they're kids, even the dose is somewhat less. Even 500 mg, they break the tablet and they give half of that is 250 mg only they provide for the kids. Okay, this is how we use the acetaminophen. Now this is not my question. Now, only I'm going to ask you a question. Like, if I want to treat fever, okay, what is the target receptor we are focusing on? For treating a common fever, what is the target receptor? Prostaglandins. Other than prostaglandins, what is some primary kind of a target? Of course, other than PG, also we have one primary target. What is that? Can someone tell me the primary target of common fever? Very good. The answer is COX-2. So COX-2 is a cyclooxygenase enzyme 2. See, whatever we are going to use the acetaminophen, it's going to target the COX-2. But what is the tissue? What is the tissue like, or what is the organ we are exactly targeting for treatment of common fever? Do you recall the tissue? Like if a patient is suffering from asthma, we are going to target the lungs. If a patient is suffering from common fever, then what exactly we are targeting? Brain. No, actually, that acetaminophen, ma'am, actually, like this, if we take more than four doses, definitely it targets the brain, because it's a slightly lipophilic drug, of course, right? But if we take more than five drugs, I mean, for even the four doses a day can create a blinding. Okay, and five doses can even kill the human. So this, of course, we should. And major suggestion to the participant: just take the positive things. The doctor can prescribe only to take three doses per day. It means that for each and 8 hours gap, we have to take that 500 or 650, whatever it targets. Okay, kidney actually, if it is a nephrotoxicity, it targets even the kidney. The brain, the central nervous, we should not target the brain and kidney. Okay, other than central nervous and nephro, what is the exact thing? Can I tell you? It looks very silly, but this is the exact answer: blood. See, how we sense the fever. We are touching our body, isn't it? So the doctor can touch or can give you a kind of a thermometer. So whatever we sense, okay, it's a like what kind of heat? Sensible heat, isn't it? In thermodynamics, we have studied about sensible heat. So the blood, in the blood only we find the COX-2 enzymes. So we are targeting the acetaminophen, whatever we provide. Even if you take 500 mg, out of 500 mg of acetaminophen, we find only 10% of API. So only 10% of API, that is active pharmaceutical ingredient. This is the exact drug we find in most of the drugs. Okay, if you find 10% of API of 500 mg, it means that only 50 milligram of acetaminophen is going to target 6 liters of blood. Great, isn't it? Just 50 mg, 6 liters. And if you see the adolescents, that is 10 to 14 age, their metabolism rate is high, just like rats and mice, very active. So the metabolism, they can metabolize even drugs within 6 hours. But if you see the general metabolic activity after 15 age, you can see it is 8 hours. So only we are prescribed to take on morning, afternoon, and night. Okay, 8 hours gap. Okay, if this is for acetaminophen for treatment of the common fever, okay, this is what a structure-based approach. See, we are discussing each and everything in detail. Now, I will give you a second example. Tell me, like, if I want to treat bowel upset. So, if I am facing from bowel upset, what is the drug you would like to prescribe for me? This is the second example I'm asking you. Or if you are facing bowel upset, what drug you ask in the dispensary to procure? Whether you go to the doctor for this, very good, loperamide. Okay, very good. So loperamide is the most preferred tablet for treating the bowel upset. Bowel, what is bowel? Small intestine. If you stretch the small intestine, it goes up to the meters, isn't it? Many meters. So that small intestine, for treating with loperamide, we take less than 50 mg, I believe. Whatever we take less than 50 mg, okay, or 5 mg, or I don't know exactly, but not more than 50 mg. So 10% of 50 mg, it comes around not more than 5 mg. That is the active drug. Okay, that is the active drug we use for treatment for targeting that intestine. Okay, so but do you know how many doses we take around? We take We have to take a single dose. There, if it is food poisoning, obviously we have to think a lot. But generally, if it is a bowel upset, a single dose is sufficient. No doctor can prescribe directly to take all the 2 doses or 3 doses, and until it is severe. So the 5 mg is targeting the 10 meters of intestines. So great, isn't it? This is what we call it as drug specificity. A small amount of drug targeting to the active sites of the protein of a particular disease. So only we are studying these drug design courses. Okay, we also have so many tools developed for the drug design, for the methods of drug design. Then what is the purpose of studying these kind of a course? So all these things you can realize when we complete the 14th session. So each and every day we are learning a lot, I believe, a voluminous information. I'm not giving a gap also. Okay, so let us see. Okay, so why we have to study all these things, you can understand the last time. So, but why I have taken you these examples. So I told you that loperamide, the residence time is more. Whatever drug we take, it is going to reside. So the drug is not going to attach like a fevicol. It's not going to permanently bind with the that acetaminophen is not going to permanently bind with the COX-2. It can reside over a particular time. That's what we call it as a drug protein binding. So if you know the drug protein binding, okay, so we can able to know the dosage levels. How many doses per day can be determined based on drug protein binding. The loperamide can bind with more time when compared to acetaminophen. So we can take a single dose per day that can able to treat the disease. Okay. So the binding affinity of ligand with receptor over a period of time can be studied using molecular dynamics. Whatever we take and we study in our human body, we are checking in computer simulation. That's what molecular dynamics simulation. Okay, so these are the softwares and outputs. We'll be discussing more about the outputs, simulation outputs, when we discuss in a separate session. Expression profiling, gene expression profiling. Okay, so this is actually a paper published actually, other than the docking. So if you see this docking, in a paper called finding the best targets for ovarian cancer, I've taken 3 important compounds and I checked with ovarian cancer. Initially, I've taken 1,000 genes for ovarian cancer, and then I screened to 100 genes, top 50 upregulated, top 50 downregulated genes. And after screening up to these 100 genes based on the pathways, then later I screened to the top 10 genes. Whatever we can see in the bottom is the top 10 genes. Whatever the top 10 genes we have taken, we have talked with the three compounds. And here you can see the negative maximum is shown from bottom to up. So here it is -6 to -8.5. And if you see the quercetin is showing a best binding energy, that is negative maximum energy of 6 targets out of 10 targets. So quercetin emerged as the best compound. Where quercetin is majorly present in our natural food. Daily we are taking quercetin in natural food. You know the name of food where we find the quercetin, more quercetin. You know the answer? Definitely, everyone are taking onion good. So onions also, but when compared to onions, tomatoes. See, tomatoes are curing the ovarian cancer. So that's what we realized after doing these computational studies. And later I checked with all the kind of papers. Whatever we found out in computational is right. Okay, so it's verified from like that. The next is pharmacophore modeling. A pharmacophore is an abstract description of molecular features. So whatever I'm showing you these methods of drug design, we'll be discussing extensively in the subsequent sessions. Okay, so no bother. When I'm going to explain these extensively, I'll be taking you to the websites, tools and databases, and I'm going to provide you the demo. Unless we know the basic purpose, no use to explain with a tool and database. Now let me explain with this pharmacophore. A pharmacophore is an abstract description of molecular features that are necessary for molecular recognition of a ligand by a biological macromolecule. So that is, if you want to check whether a ligand is going to bind with a receptor or not, that's what we call it as docking, isn't it? But here there is a small change. In docking, we generally assess through docking energy, binding energy. Here, we assess the binding of ligand with the receptor using fitness values, fitness factors. Okay, we use a different kind of a correlation and we check the interaction of functional groups of a ligand with uh, the receptor. Okay, so this is something about pharmacophore. And then pharmacophore modeling is before talking, we have to make this kind of a pharmacophore modeling where a ligand has a lead backbone structure. Whatever lead backbone structure we have, okay, we can attach and detach the functional groups. And then we can bring a new compounds. And these compounds can also check, we can check binding with the target receptor, and if we find a good affinity, we can take that as a base drug. Of course, pharmacophore modeling can be used for this purpose. For pharmacophore, we take pharmacophore features like hydrophobic centroids, aromatic rings, hydrogen bond acceptors, donors, cations, and anions. So these are the major molecules we take for assessing the hydrophobic and hydrophilicity of ligand over the receptor. So we also have steps similar to that of QACR. So more about this we discuss on the steps. And the pharmacophore is an abstract description of molecular features that are necessary for molecular recognition of a ligand by a biological macromolecule. So whatever ligand you take, you first check with one particular target and then we assess it. Okay, so for this we can use a tool called Gene Farmer. A compound called bicalin I tried as an input and I got this kind of a result. Okay, these are a few more softwares to work with for mapping. There is also one more tool called ForMapper. If we can able to upload the structured data format of a ligand, okay, then obviously we get series of target receptors, something equivalent to target deconvolution. But one thing you see here in this online tool, we can get the fit score. Okay, so instead of binding energy scores, we get something like fit score. Based on the ascending order, you can see. Okay, this is the nothing is described, So only thing is that you have to go to the PDB ID to check the name of the protein. But the second best protein is the DNA-directed RNA polymerase. That is, it can be used to treat RNA viruses. So this shows an antiviral activity. Like this, we can check our ligand affinity over different receptors. So this is of course a part of a product. I mean, part of a output of the FarmMapper. PSR, I think we have already discussed. So we have discussed the major theoretical information in drug design. So on tomorrow we'll be discussing much of the hands-on session. So we'll be compensating the much of practice of online tools and databases on tomorrow. Okay. So that is on tomorrow. We'll be discussing on drug databases, drug likeness, and ADMAT, and also hands-on sessions. Any doubts you can ask me at present. Today I've completed the session. Presently, I can end with a quote: "Half knowledge is always dangerous." You accept this, isn't it? Abhimanyu, Arjun story. So uh, try to attend the subsequent sessions. Okay, definitely we can keep in. You can understand the difference between the different methods and applications also. Yeah, you can ask me the question if you have any doubts.
Namaste, sir. Actually,
Uh,
I'm a computer engineering student and faculty. So, sir, the problem is, can we get some good reference book also alongside so that I can clear certain of the
uh,
doubts in that
okay.
context?
So
Because, yeah,
there is one book called Molecular Modeling by Anand Solomon. Okay, so this book, sir, Molecular Modeling by Anand Solomon. Here you can find all these drug design methods. Whatever I mentioned, this is a very small book, thin book, but this Anand Solomon has written well with all these kind of a concepts. If you want to follow the textbooks, I suggest you to go with this or Medicinal Chemistry by Gareth Thomas. So in your library, if you can find these books, medicinal chemistry, because a chemistry division definitely will be there. If you focus on these books, it can give you much knowledge. And sir, actually, these are very easier to use. No need to go with a complex programming. Myself, I program many games and many softwares, but I'm not popular at that time. But using the existing softwares, I'm getting more reputations. So even though it is new for you, you can learn this kind of application of tools and databases using these books, whatever I mentioned. So, and other than this, the contemporary researchers can be learned through journal papers. Soon, I'll be circulating the published journal papers. If you follow my journal papers, which is more related to our course, you can understand well. Any other doubts?
thank you, sir. Thank you.
Thank you, sir. So if no doubts, and even if you accumulate any doubts, we can see it on tomorrow. Okay. Have a great day to you all. Thank you for your interaction. Today you have interacted well. So I thank you for that and also for maintaining the silence. Have a great day to you.